So now let us continue with the next stage of human reproduction, that is the process of fertilization. And fertilization is the union of the male and the female gametes, where the male pronuclei and the female pronuclei come together, result in the formation of the diploid zygote. फर्टिलाइजेशन के टाइम पे पहले तो मेल और फीमेल गैमेट्स यूनाइट होते हैं, जिसको हम लोग सिनगैमी कहते हैं। उन दोनों गैमेट्स का साइटोप्लाज्म मिक्स होता है, जिसको हम लोग प्लाज्मोगैमी कहते हैं। उसके बाद ये स्पर्म और ओवम के प्रोन्यूक्लियाई एक दूसरे के साथ फ्यूज हो जाते हैं, जिसको हम लोग कैरियोगैमी कहते हैं। क्रोमोसोम्स का इंटरमिंगलिंग होता है, जिसको हम लोग एम्फेमिक्सिस कहते हैं। तो जब मेल और फीमेल गैमेट्स यूनाइट होते हैं, उसको सिनगैमी कहते हैं। उनके साइटोप्लाज्म के मिक्सिंग को प्लाज्मोगैमी कहते हैं। मेल एंड फीमेल प्रोन्यूक्लियाई के यूनियन को कैरियोगैमी कहते हैं। क्रोमोसोम्स के मिक्सिंग को हम लोग एम्फेमिक्सिस कहते हैं, ultimately resulting in the formation of a diploid zygote. The site of fertilization is the ampulla. The sperm enters after insemination, but the sperm is covered by some cholesterol vesicles, which will prevent the entry of the sperm into the ovum. So they need to be removed. That process is called as capacitation. So now the sperm will become active. The sperm sperms start vigorously moving towards the ovum. The ovum shows the presence of layers called the corona radiata, zona pellucida, the vitelline membrane. So it goes like this. There is a vitelline membrane, which is actually the egg membrane, followed by the layer that is called as the zona pellucida. It is a non-cellular layer, followed by the corona radiata cells. This forms the egg. And you have the female pronuclei with its ooplasm. यहाँ पे ZP3 रिसेप्टर्स रहते हैं। स्पर्म्स के पास उसकी कॉम्प्लीमेंट्री प्रोटीन्स होती है, जिसकी वजह से स्पर्म्स अटैच हो जाते हैं ZP3 रिसेप्टर्स के ऊपर. So as a result of which few hundred sperms they bind to the zona pellucida and they start releasing zona license or acrosins, which try to penetrate the zona pellucida. Okay, as the sperms penetrate the zona pellucida, one of the sperm will touch the vitelline membrane first, and this is the syngamy. Immediately, the ovum will engulf the sperm inside, and as the sperm gets engulfed inside, this nucleus of the ovum, the secondary oocyte, which was arrested at meiosis 2, will complete its meiosis 2, resulting in the formation of the female pronuclei. Already यहाँ पे मेल pronuclei आ गया है. तो ये इवेंट्स बराबर से समझिए. अब ये जो विटलाइन मेम्ब्रेन है और जोना पेलुसिडा है, यहाँ पे the vitelline membrane will undergo a reaction resulting in the formation of a fertilization membrane so thick that no other sperm will be able to enter into the ovum now, and that will prevent polyspermy. Ab under male pronuclei, female pronuclei, both the pronuclei will unite and they will result in the formation of the diploid zygote. So that is how the zygote formation will occur. Now the zygote will undergo cleavage, which is slightly different from the regular mitosis. You can refer our class notes for that. The zygote will undergo mitotic divisions. The first cleavage So type of modified mitosis will result in the formation of two blastomeres 30 hours after fertilization. Second cleavage will result in the formation of the 4-cell stage. Third cleavage will result in the formation of the 8-cell stage, ultimately will form the 16-cell stage. And this 8 to 16 cell stage is called as the morula stage. Now the cells of the morula, they get rearranged, result in the formation of an outer layer of cells and an inner layer of cells. These cells, when they reach the uterine wall, they absorb the uterine fluid, result in the formation of the blastocyst, जहाँ पे outermost layer of cells को हम कहेंगे trophoblast, and the cavity that is formed is called as the blastocele, and the cells get attached to one layer of the trophoblast. These are the inner cell mass, or it is called as the embryoblast. And this embryoblast will actually form the fetus. It will form the embryo first with the ectoderm, mesoderm, and the endoderm. The trophoblast helps in the formation of the extraembryonic layers. Implantation के टाइम पे the uterus which secretes the uterine milk absorbed by the morula, blastocyst वगैरह फॉर्म हुआ. अभी तक zona pellucida is retained because now the zygote was traveling from the ampulla up to the uterus. It's a very sticky structure. You don't want it to stick to the other parts of the fallopian tube or the wrong parts of the uterus, or an ectopic pregnancy will begin. Pregnancy will begin at a wrong site. So to prevent that, the zona pellucida is retained. When it reaches the uterine endometrium, the zona pellucida is removed and the blastocyst undergoes implantation almost on the 7th to 8th day after fertilization, and that will result into the pregnancy and the gestation will start. During implantation, the trophoblast has a very important role to play. The trophoblast results in the formation of these villi which penetrate through the endometrium. वो लेयर को हम लोग सिंक साइटोट्रोफोब्लास्ट बोलते हैं. Then the next stages है वहां पे we get the formation of the three germinal layers, and the formation of the three germinal layers is called as gastrulation, where we get the formation of the ectoderm, mesoderm, and the endoderm.